Good morning, everyone. Welcome to the Arthritis Web Seminar Series. Today is our lecture number 43. Our guest speaker today is Dr. Rick Loris, and he's going to speak on molecular identity of the articular chondrocyte to prevent and treat osteoarthritis. Before we start the introductions, just a reminder to the audience, please post your questions on the Q&A tab on Zoom. Rob?
Good morning, everyone. It's a personal delight to welcome Rick Loris to our web seminar series. I've had the good fortune of knowing Rick over many years. Our paths have crossed in many circumstances. Rick is a full professor and head of the Department of Development and Regeneration at KU University in Leuven. His lab includes a focus on tissue homeostasis and disease lab, which he directs. And he's also chair of the Division of Rheumatology at the University Hospitals in Leuven. Rick's had a long-standing interest in axial spondyloarthritis, osteoarthritis, and PSA, and doing cutting-edge translational research in a very innovative and creative way with high-impact research publications over a long period of time. He's also had an outstanding record of leadership, both in ORSI and the Belgian Society of Rheumatology. He is on the board and played a very active role in publications, including a role as associate editor for Annals of Rheumatic Disease and Osteoarthritis of Cartilage. So Rick, it's a delight to have you here today. Thanks for joining us. I'll pass it over to you.
Thank you. Thank you for the kind invitation to participate in this prestigious series. I'm on the other side of the Atlantic, so I have to talk to you from this nice medieval town that features two things: an old university going back to the 16th century, and the Stella Artois brewery, which has expanded into the world. It is now a leading craft beer in most continents. As you can see, we have a historical center, we have a vibrant nightlife, we have the nice things to eat, for which Belgium is very well known. And like you in Toronto, we like sports. We do not have a ice hockey team, we don't have the ice, so we have a field hockey team. And it happens that by coincidence, by scientific approaches, that Belgium is currently European, world, and Olympic champion. Yet there's another thing that we share with Toronto. We like bikes. You have the Cervelo factory that makes race bikes, and everybody in Belgium is genetically predisposed to love bikes, so everybody knows the subtypes. And we were happy to host the World Championships last year, which created the Stanley Cup-like atmosphere in the study. So I hope to convince you that someday, if you have not yet visited us, that you consider us on one of your European destinations. That being said, I will talk today indeed about a subject that I care about a lot and that I think provides unique opportunities to develop therapies for osteoarthritis, and that is knowledge of the molecular identity of the articular chondrocyte, a unique cell in our body. As introduced by Rob and Mohit, I work both in the clinic and in the lab, and we have a big clinical department in an academic third line hospital, as well as a big research center. And so in that context, my disclosures are that as a team and personally, we work together in consultancy and research with a number of companies, of which two, Galapagos from Mechelen, Belgium, and Biosplice, formerly Semamed from San Diego in the United States, are trying to put products on the market to treat osteoarthritis. Above that, we are particularly grateful to the funding organizations that support our work, both at the national level as well as at the European level. And without their money, we couldn't do the research. So who are we at the Skeletal Biology and Engineering Research Center? We're a group of people that tries to understand in a very broad way cellular and molecular basis of skeletal tissue formation, homeostasis, disease, remodeling, and repair, including tissue engineering. So we eye in the long term to develop all kinds of therapies for a number of skeletal disorders, and the main ones that are in our focus today are osteoarthritis and bone healing. We are a multidisciplinary team, and much of the work that we do would not be possible without the input of different people with different backgrounds. You can see the academic staff down here. It features Professor Monteagudo, who is a pharmacist and who is co-directing the tissue homeostasis and disease lab with me. And then we have a number of engineers, Lies Geris, Yannis Papantonio, Christa Maas, and Veerle Bloemen, who work more in regenerative medicine, but also in developmental biology. And we have Ilse Jonker, Lucas, who's a physiotherapist, and together with Lise, she's a master in modeling of molecular events as well as movement and disease. So that brings me to our main common interest that we have in the SBE Research Center, and that is osteoarthritis. Now, it would be way too simple to say that osteoarthritis is a simple disease. It's not a single disease, by the way. It's probably a common outcome of a number of different disease processes. But what is important, of course, from the clinical perspective is that osteoarthritis is associated with a significant burden in terms of pain and loss of joint function, and that we currently do not have any effective therapies, at least not for the long-term prevention of further degeneration of the joint. We know it's a multifactorial disease to which mechanical factors, systemic factors, and genetic factors contribute. And we have found over the years that studying genetics of osteoarthritis is quite an interesting way to identify targets, to identify pathways that are involved in the pathophysiological processes, and then take this on further forward in translational research approaches. We know today that osteoarthritis is definitely not a simple degenerative disease. It's not a natural consequence of aging. It's not the natural outcome of any joint trauma that you may have. No, it's a complex disease to which the different tissues of the joint all contribute. And that includes changes in the articular cartilage, includes changes, inflammation in the synovial tissue, leading to synovitis and effusion in the joint. It includes changes in the subchondral bone. It includes changes in the bone marrow. So it's really a disease of that whole joint organ, but we do think, at least from the perspective of the research that we do, that the articular cartilage is a very prominent tissue. And that is our main field of study. We, over the years, have developed quite some insights and strategies and concepts that we try to apply to the health, the homeostasis of articular cartilage, and then subsequently what happens in the disease. And as a team, we are convinced that this is a way, not the way, but a way to go to find better solutions for the disease as it is lived today. So articular cartilage, you will probably all be very well aware of this, is a unique tissue in our body. It's cells and extracellular matrix. The cell population is unique. It's the articular chondrocyte, but we do know also that within that population of articular chondrocytes, there is some diversity depending on the place within the joint where this specific cell is found. And so the upper layers are very important for lubrication of the joint and to deal with the shear stress, whereas the deeper layers are more important to deal with hydrostatic pressure upon movement of our joints. Now, the fact that we have a unique cell type within that tissue and that we largely have a non-vascularized tissue makes this a very interesting tissue to study, and obviously with impact on important disease processes. Now, the articular chondrocyte is not the only chondrocyte. There's another type of chondrocyte that is also very interesting to know about, to understand the biology of, and that is the developing chondrocyte. During development and growth, stem cells differentiate into chondrocytes that subsequently go through a series of different differentiation steps, becoming pre-hypertrophic and subsequently hypertrophic. And these steps are essential in normal bone formation, are recapitulated to some extent in fracture healing, and also in the pathological new bone formation process, as we can see in axial spondyloarthritis. The important thing is that these hypertrophic chondrocytes, which represent a terminal differentiation state, likely go towards cell death, most of them, although some may transdifferentiate to osteoblasts, but at least they disappear and they make room, they make space for new bone formation to occur. And that's the way we develop our long bones. Now, the difference between the articular chondrocyte and the developing chondrocyte is quite important if you start thinking about what is happening in osteoarthritis. This is one of my favorite books because it features nothing but nonsense, which is very good if you look at something late at night. And like the author says here, there's two options, and you have two types of chondrocytes. So you have the hypertrophic chondrocytes that work hard and die poorly. They even have to become osteoblasts, some of them. And then you have the articular chondrocytes that work smart. They make their extracellular matrix, and then they feel like they've done the job. They retire and they just rest and watch their extracellular matrix being renewed at a very slow pace. So it's like an eternal holiday. The only thing that the articular chondrocyte did not take into account was that whenever something is happening to the joint, such as a trauma, that they are not well equipped to deal with this in the proper way, and that they show a panic response. And that is basically what we have documented with many teams over the last 3-4 decades, is understanding molecularly what is happening with these cells in the process of osteoarthritis. And it seems like the cells are losing their common sense. They act program when they are being stressed. With cell proliferation, they increase their matrix synthesis, which is a logical thing to do if the matrix is damaged, but at the same time start producing lots of pro-inflammatory cytokines, producing tissue destructive enzymes, and they lose that stable normal phenotype, and they become, at least part of them, more like the hypertrophic chondrocytes that we know from developmental biology. And this is a bad process because the production of molecules such as vascular endothelial growth factor and such as MMP13, a major joint extracellular matrix destroying enzyme, are bad events that will lead ultimately to thinning of the articular cartilage and to the cells being in another type of extracellular matrix that is biomechanically inferior and thus contributes to the more rapid decay of the joint. So if we can manage to influence that molecular identity and help the cell, teach the cell how to react in terms of a challenge, we may come a big step forward towards better treating a disease like osteoarthritis. Now, we hypothesized quite a while ago that maybe the Wnt signaling pathway was a pathway that is critically involved in this. And effectively, over the last 10 years, a number of groups have all contributed to our understanding of Wnt signaling in the context of joint biology. And what we know now is that basically every cell in the joint can be sensitive to Wnt signaling and mostly reacts, if you look at it from the perspective of osteoarthritis, in the wrong way. We know that increased Wnt signaling is associated with increased bone remodeling, with the bone sclerosis phenotype, with synovitis, with cartilage degradation, with chondrocyte hypertrophy, and therefore less optimal extracellular matrix and rapid decay of the joint. So thinking about Wnt signaling is showing, all these concepts are showing us that we actually may have a potential therapeutic target there. And effectively, we know that on the surface, there is a little amount of Wnt that remains essential for the health of the superficial layer, but in the deeper layers that you need to control the extent of the activation of that pathway in order to avoid this deleterious cascade of chondrocyte hypertrophy. And actually that work started a long time ago during my postdoc when we studied a new knockout mouse, which was a knockout for a molecule called secreted frizzled-related protein, or FRISB, and that is a secreted Wnt antagonist. A couple of years before that, John Laughlin and Maribeth Kaur from Newcastle and San Diego had suggested by a genome-wide study that there may be links between polymorphisms in the gene encoding FRISB and osteoarthritis, and we were at that point developing a new knockout mouse, mainly for developmental biology use, but it turned out these mice did not have a phenotype, and we subsequently started to challenge them in different models of osteoarthritis, and what we found is that in the absence of an extracellular Wnt antagonist, that there was more severe disease, thereby providing actually the first evidence that a hyperactivation of this signaling pathway would lead to disease. Now, very short on the signaling pathway, the typical Wnt signaling pathway is focused on a molecule called beta-catenin. Beta-catenin is normally a cytoplasmic molecule, and when the Wnt binds to its receptors, which features a frizzled receptor and a core receptor, beta-catenin will be stabilized in the cytoplasm. It will accumulate and go to the nucleus. In the nucleus, is associated with very specific transcription factors and influences the transcriptional response. Under normal circumstances, when the cell does not see a Wnt signal from the outside, what happens is that beta-catenin is caught in what we call a destruction complex, multi-protein complex, leading to the ubiquitination of the molecule and its breakdown in the proteasome. So upon activation, you get the opposite effect. So in the absence of Wnt signal, beta-catenin disappears. In the presence of Wnt signal, it is there and it will go to the nucleus. And shortly after the paper on FRISB that we published, the team of Regis O'Keefe, who was back in those days in Rochester, New York, but is now in St. Louis, demonstrated that both the absence as well as the as a functional increase in beta-catenin led to an osteoarthritic phenotype. And that was quite interesting because it shows that Wnt signaling need to be tightly regulated within the joint for the joint to remain healthy. If you get rid of the function, the chondrocytes die. Basically, you get a decay of the cartilage following that of the chondrocytes. If you overactivate the pathway with a constitutively active beta-catenin that is stabilized and going to the nucleus, what you get is chondrocyte hypertrophy and progressive osteoarthritis in mice. So this is definitely a very interesting pathway if you look at it from the perspective of the molecular identity of the articular cell. We now know that the pathway is very complicated because it needs fine-tuning. There are a lot of regulatory mechanisms, and many of these regulatory mechanisms can actually be used to have some form of pharmaceutical interference with it. And this is summarized in this slide. You can basically work on inhibiting Wnt secretion. Wnt are glycoproteins with a lipid tail, which means that the lipid has to be attached to the Wnt. So this is a process that is taking place in the ER and in the Golgi apparatus, and you can technically interfere with that. You can interact or you can interfere with Wnt receptor binding. You can work on the co-receptors. You can work within the cell on the different members of the destruction complex, of which many have enzymatic activities. You could go and try and work deeper in the cell on intranuclear targets, looking at the transcription factors, and one of the drugs that is in development, which is called lorcivivint and is being developed by BioSplice, is a molecule that affects differential splicing, among others, of the Wnt transcription factors. So there are really different levels where you can achieve success. But the big question is, can we actually target this pathway in a safe way? And that question is being asked because Wnt are really what we call morphogens. They can impact cell fate decisions, they regulate differentiation, they regulate proliferation of cells. And abnormal Wnt signaling, mostly excessive Wnt signaling, has also been associated with a number of tumoral processes. And actually, the Wnt were originally discovered in that context. So despite the fact that there are many diseases where one could target this, on the other hand, many of the companies and many of drug developers are a little bit scared and apprehensive that the broad effects of this pathway may interfere with safe targeting. And that probably means that we need to go for specificity, preferably tissue specificity, either by the way of administration or by defining the targets. And we have learned over time now that specificity of the tissue in terms of administration can, of course, be achieved by intra-articular injection. Now, after a number of years ago, there was another molecule that we discovered in collaboration with geneticists from Rotterdam and many other teams, and we contributed to the functional evaluation of that, which is called DOT1L. And DOT1L is what we call a histone methyltransferase. The histones are the proteins around which our chromosomes are wrapped, and they're very important from the epigenetic point of view. Histone modifications can free up or can lock pieces of DNA in order to influence transcription. And so histone methylation, like histone acetylation, is one of these processes that helps to fine regulate the transcriptional machinery. And DOT1L is quite a unique molecule because it's the only known H3K79 methylase, which means methyltransferase, which means that it methylates the lysine 79 group on histone 3. This has been associated with a number of processes during development as well as with disease. And so in collaboration with our friends from Rotterdam, we found a functional association between polymorphisms in DOT1L and osteoarthritis. And subsequently, we went on to further study the biology of this. This is probably a good moment to introduce the most important members of the band. Much of this work was done by Silvia Monteagudo, who was at Dutch Times a postdoc in my team and currently is the co-director of my lab. Most of the mouse work, including the generation of new transgenic animals, is being done by Frederik Cornelis, a senior postdoc in our team with plenty of experience in mouse transgenics. And then Fred Cayoto, if you think this guy may look like a French guy, you're absolutely correct. He is from France. He was with me as a postdoc for 5 years and has now returned for his own, and has his own lab in Nancy. So these people contributed mainly to this study, in which we showed that actually DOT1L is regulating Wnt signaling in the context of osteoarthritis. We used the chemical inhibitor. We demonstrated that with that chemical inhibitor, we could interfere with the activity of the methyltransferase, leading to a reduction in the levels of H3K79 methylation, and functionally, when we did that injection intraarticularly, to the development of osteoarthritis in the knee joint. So basically, active H3K7 active .1L protects against osteoarthritis. And then we found out doing microarray analysis as well as functional cell experiments that, in fact, if you interfere with the function, if you delete the gene or if you interfere with the function of .1L, that upon Wnt stimulation, you get a true hyperactivation, suggesting that .1L is acting like a brake on the activation of the signaling pathway. And we could take that further forward to an in vivo setting. We could inhibit the effect or we could counteract the effect of the .1L inhibitor by giving a Wnt inhibitor. And we further figured out how this works more in detail in the protein complexes at the level of the chromatin. Now, all of this shows more or less that Wnt signaling is really regulated at all levels of the signaling cascade, and that provides opportunities for drug development. But the deeper you go, the more opportunities we may have for specificity. And .1L was our first example to show that beyond classical approaches, like for instance an extracellular Wnt inhibitor, that there are maybe other ways to interfere with the pathological processes that we see in a way. So and that brings me to the next perspectives. Based on the earlier observations, I told you about the genetic association. I told you about the Wnt hyperactivation as mechanism underlying the development of OA with a loss of function of .1L. And then we had a third study in which we developed transgenic animals in which .1L was absent in cartilage, once again leading to OA. We came to the conclusion that it would be interesting to know how .1L itself is being regulated, because we noticed that within OA samples, not only the amount, not only the function, but also the amount of .1L appeared to be reduced, as well as its downstream products. And this is work mostly done by PhD researcher Astrid de Rover in our team. She developed a bioinformatics pipeline that looks quite complicated, but basically uses a number of different prediction tools for transcription factor binding to genes, then applies an algorithm that looks for specificity, that looks for shared information from different bioinformatics tools to functionally end up with a list of transcription factors that are likely to influence the expression of the molecule. And we took that together with our RNA-seq data from the cells in which .1L was inhibited. And the bioinformatics analysis suggested that the response to hypoxia was probably an important factor in the regulation of .1L. And indeed, in some of the networks that we have drawn, HIF1 alpha, which is a key mediator of hypoxia as a transcription factor, was right in the center. Looking more specifically in detail, indeed, we find hypoxia HIF1 alpha binding elements in the .1L promoter. Now that may be important in the context of OA, because actually the articular cartilage is largely a hypoxic tissue. I told you it's non-vascular, so you can guess that the oxygen gradient is low. And actually the gradient or the amount of oxygen goes down from the surface of the articular cartilage towards the deeper layer. So in the deep depths where the main bulk of the cells are that make up the extracellular matrix that gives the function, the oxygen amount is very low. On the other hand, we also know that oxygen levels are dysregulated and that hypoxia appears to be lost when osteoarthritis is developing, both in mouse models as well as in humans. So we went out, or Astrid went out to do a lot of functional experiments to really define to which extent indeed hypoxia is key in controlling .1L expression and activity. And so using a cell line, she demonstrated using different hypoxia, inducing hypoxia or a mimic of hypoxia in different ways, that effectively upon the creation of a hypoxic or a hypoxia-like environment, that the expression levels of .1L go up and parallel those of a well-known target of HIF1, which is VEGF alpha. So we did this both chemically with inhibitors of different enzymes that are involved in the stability of HIF1, creating an hypoxia mimic-like situation. But we also did this in a hypoxia incubator in which the cells were kept at very low oxygen. This also occurs at the protein level, and if you look at the left of the screen, you can see that upon exposure to low oxygen levels, the cells upregulate HIF-1 alpha, which is the essential transcription factor in this process. This leads to higher amounts of the enzyme DOT1L, and it leads progressively to higher amounts of the products of the enzyme, the histone methylation at H3K79. And functionally, that is further translated into a better quality of cartilage. What's the underlying mechanism? As I showed you, there are different binding sites for HIF-1 alpha in the DOT1L promoter. So we made a number of promoter constructs with and without the binding site. And what we could show is indeed that in the presence of hypoxia, that there is more luciferase promoter signal, indicating that we are dealing here with direct binding. And this has functional consequences, and it's fairly unique for HIF-1 alpha. We did not see that for the family member HIF-2, and we could use chromatin immune precipitation, as shown on the right side of the screen, to show that it really binds so that the physical interaction is direct. Does this have any further meaning? Well, we tested this in vivo using osteoarthritis models, and this is a model that you are probably well aware of. If you're working in the field, we used the DMM, the destabilization of the medial meniscus model, and as a therapy, we injected these mice over the course of the experiment with IOX2, which is a chemical hypoxia mimic. It stabilizes HIF-1 alpha, leading to a transcription program that is similar to the one that you would see when you put the cells under low oxygen conditions. And these were the results. What we could demonstrate is that in the presence of IOX2, that by recreating a more hypoxia-like environment, that the articular cartilage was protected when performing a histological analysis using the Orsi score. Quite interesting, but is this also linked with indeed with DOT1L activity and with the DOT1L product? And effectively, not only did we see it at the level of the articular cartilage, we also saw it at the level of the osteocyte, of the forming osteophytes. But moreover, when we did a number of histological stainings, we could see that not only the levels of DOT1L went up and the levels of HIF-1 alpha went up when we treated with IOX2, but also the product of the enzymatic activity of DOT1L went up, and we saw more H3K79 methylation. And see the difference on the right side of the graph when you induce the DMM model and you develop osteoarthritis in the mouse, the levels of the methylation of H3K79 really go down, and we can restore this by giving the hypoxia treatment, showing how powerful this effect is. Now, for the final part, I'll go to another molecule, which more or less follows the same tracks, but came with a lot of surprises. We demonstrated in 2018 that a small molecule that is ubiquitously expressed called AMP32A is essential in reducing oxidative stress in many tissues and in many aging-related disorders, including osteoarthritis, osteoporosis, but also in some neurodisease. And actually, we started working on AMP32A back in 2007, leading to a paper in 2009 in one of the very first genome-wide association studies that was run by Anna Valdez and Tim Spector. They found a potential location close to the AMP32A gene, which gave some signal. And subsequently, Anna went on to look in more detail at variation of this gene, and we did some expression studies in the articular cartilage, and we found that there is probably a genetic association or a higher risk with variants for AMP32A and hip osteoarthritis in women. And then it took us about 9 years to come up with a conclusion, and a lot of work and a lot of mouse analysis and a lot of bad moments and a lot of high moments in the lab. And what we demonstrated then in 2018 is that AMP32A plays an essential role in our defense against oxidative stress, because it is absolutely essential to maintain the expression of the ATM gene, the ataxia telangiectasia mutated gene, which is an important regulator of our antioxidant defense. So in the absence of AMP32A, you would get excessive oxidative stress leading to osteoarthritis, to cerebellar ataxia, and to osteoporosis. Now, what is interesting is that we had a very simple way to correct the osteoarthritis in this genetic mouse model. Lots of oxidative stress. The response to that is give an antioxidant drug, well-known N-acetylcysteine, that we could just give by the drinking water. And this was largely capable of restoring the integrity of the articular cartilage in this mouse model, which we found quite exciting and which we then extended also to, for instance, the cerebellar ataxia phenotype, which we could also correct simply by administering an antioxidant drug. Yet we felt that we did not cover the full range of AMP's regulatory functions because there were some other signals coming out of some of the analysis that we performed. Because when we were looking at what was happening in the mouse model, we did microarray analysis in those days of the articular cartilage, the AMP32A knockout mice. And when we went more in depth, finding which pathways, which cascades could be really regulated by AMP32A, we found that beyond everything that has to do with oxidative stress, all of a sudden also Wnt signaling appeared on our radar. So we hypothesized, well, if AMP32A has an impact on Wnt signaling, how would that work? And for that, we used a model of micromasses using ATTC5 chondrogenic cells. If you culture them in micromasses or in droplets, they will undergo, with the right cocktail of growth factors, a process towards chondrogenic differentiation, including chondrocyte hypertrophy and finally mineralization. And in those cells, we genetically silenced AMP32A, mimicking thereby a process that results in loss of function. And we found that under those circumstances that Wnt target genes were upregulated, different ones. And we found that the absence of AMP32A in the differentiation model had a strong impact on the chondrogenic differentiation of the cells. As you can see here, there is a strong decrease in collagen type 2, in aggregan production, as well as in collagen 10 production, and in the accumulation of extracellular matrix, as you can see from the micromass culture pictures. So it seems like AMP32A, or the loss of AMP32A, upregulates or activates or enhances Wnt signaling, and this goes hand in hand with deleterious effect on the gene expression program of the articular chondrocytes. And effectively, at least for some of these genes, like aggregan and for collagen type 10, by using a Wnt inhibitor, we could restore the normal phenotype, linking both pathways with one another. We went to mouse. Again, we looked in the AMP32A knockout mice. We looked at the cartilage. We found that in addition to excessive oxidative stress, we also found evidence for an upregulation of different Wnt target genes. We went a few steps further. We did DMM surgery. We let the mice age, and in every single circumstances, we noticed that the number of cells that were showing activation of the Wnt signaling pathway were higher in the knockout mice compared to the wild types. So maybe AMP32A is also a factor that contributes to the regulation of Wnt signaling, and the absence would lead, just like the absence of DOT1L, to too much, and too much, as we already know, is a bad thing. Any translational evidence for this? Well, we noticed that within knees and hips of patients with OA, that the levels of AMP32A were much lower in the damaged areas compared to the preserved areas, and that they were also lower in the osteoarthritic knees as compared to the healthy knees. And in this context, we found a strong correlation with the expression of Wnt target genes, but in the opposite way. No AMP, high Wnt activation, high AMP, low Wnt activation. How does it work? Well, AMP32A is a cytoplasmic protein. We found that it's actually part of the destruction complex because it binds to axin and in that complex. But upon activation of the pathway, it gets disconnected from the destructive complex, and it attaches to beta-catenin, travels together with beta-catenin to the nucleus, as if beta-catenin is carrying a natural break in the system. Because in the presence of AMP32A, the response towards Wnt signaling is reduced as compared to the absence. So we thought, well, we touch here upon a new internal regulatory system that can limit the hyperactivation of Wnt signaling, which, as I told you earlier, is something that we consider a deleterious event. One step further, we found once again that actually this function of AMP32A is also an epigenetic regulatory function, because what we see is that by interacting in these transcriptional complexes, it has a masking effect on histone acetylation. And for many of these target genes, histone acetylation is a way to activate the gene transcription epigenetically. So if you have a molecule that interferes with this process, then you have something which will have the repression, and that is acting kind of this, what I call the brake function on the hyperactivation of the signal. Does this have any further clinical implications? I already told you that these mice have excessive oxidative stress, which we can counteract with N-acetylcysteine, but they also have Wnt hyperactivation. So can we do something with the combination of both strategies in the context of a mouse model? And that's exactly what we did using, once again, the DMM mouse model. We combined treatment with a Wnt inhibitor with that of the antioxidant molecule and N-acetylcysteine. And we showed that actually the effect is most outspoken, or the effect on cartilage damage is almost totally dependent on the NAC, on the N-acetylcysteine in this specific model. Remember, this is a genetic model in which AMP32A is absent, and it has really high oxidative stress. Yet, if we look further, the effect on osteophyte formation that we have is more dependent on Wnt signaling. So if we inhibit Wnt signaling in this specific genetic model, we have a negative effect on osteophytes, suggesting at least that combination of two strategies can do something about the multiple phases that osteoarthritis has. Once again, any impact on further biology, we looked using the Human Protein Atlas database for systems in which there would be high levels of AMP32A and low levels then of activated Wnt signaling. And effectively, we found these in muscle and brain, and among the muscles also features the heart. And just as an example, we looked at the effect of the absence of AMP32A on the heart of mice. Again, we found hyperactivation of Wnt signaling associated with high levels of the target gene TCF1 and associated with remarkable hypertrophy in these mice. What you can see here is a macroscopic image of the heart and then some microscopy. I think you can clearly appreciate the hypertrophy effect. So with AMP32A, we have another player that not only regulates the antioxidant response, but contributes to the maintenance of the molecular identity of the articular chondrocytes by regulating Wnt signaling. And once again, this is something that is not only happening in cartilage, but also in a number of other tissues. And our way forward today with the research, both with DOT1L as well as with AMP32A, is further defining the factors that regulate AMP32A in cartilage. And we will hopefully shortly be able to submit a manuscript in which we used a similar bioinformatics analysis as the one for DOT1L, showing that again, hypoxia, but also Wnt signaling itself in kind of a loop are the factors that control the expression of AMP32A in our articular cartilage setup. So what do we want to do in the next couple of years? We want to continue looking at the intracellular regulation of Wnt signaling in the chondrocyte identity, focused on hypertrophy, and we have a number of other systems in place. We're also performing or have performed RNAi screenings on a large scale to find further mediators of DOT1L and AMP32A. We're looking at single cell sequencing data and epitope studies to better understand this molecular identity. And we are taking a new adventure doing more work on metabolism and including lipidomics of chondrocytes, because we are starting to find some links between lipid metabolism and the health of the chondrocyte. And we are not forgetting that after all, the joint is a biomechanically stressed environment, that we have to deal with the loads, and we're trying to further integrate, and that's the advantage of a multidisciplinary center, biomechanical signaling and biomechanical modeling with our molecular tools. So just to give you a glimpse of the future, what we have now with Yannis in the team is a platform to create microaggregates of stem cells differentiating into chondrocytes, making organoid-like structures that we can use not only for their favorite tissue engineering approaches in the healing of large bone defects or of osteochondral defects, but also as in vitro tools, thereby trying to replace some of the mouse work or replacing doing something about our high need of having primary human articular chondrocytes, which of course come on a limited basis from clinical practice. We also have invested a lot into the virtual world. If you go to bioRxiv, you can find the single cell atlas of the skeleton that has been developed by Tim Herpeling, who's a student of Lee Scheres in the team. But we take it one step further. The models that these engineers can make with the in silico tools help us to predict experimental outcomes, which we can then validate in a restricted number of experimental settings, helping us save time, but also helping us develop new initiatives and new hypotheses. This is a little bit of workflow that we have today, of which I'm really happy, because we're trying to integrate that with our more traditional translational approaches. We can biomechanically model a knee joint, we can biomechanically model the articular cartilage itself, we can go to the cell level, and then we have all the data from transcriptome analysis, and we're trying to add to this epigenetic analysis to make molecular networks that we can in silico manipulate to see what is predicted to happen with the biology, and then take this forward towards a more translational context, replying to some of the research questions that we have. So that brings me more or less to the end. I hoped to convince you that among the many pathways that are involved in the biology of the joint, that the wind signaling pathway is certainly a beautiful one to think about in the terms of pathology. And at the same time, I also wanted to highlight that I'm lucky to be working in a center and with people that bring many different dimensions to the field, and that we hope to make further progress. Showing you the better sides of life in our lab life. This was mostly before COVID, but things are returning. So I hope that just like you, that we will benefit from all sides. And I will close this one just by advertising something, if Mohit lets me do that. We do have currently an open postdoc position for epigenetic factors in OA. And if you're interested, you can email to me or to Sylvia. Thank you.
That's terrific, Rick. Wonderful talk. We have a number of questions popping up on the Q&A tab. So first question is by Jason. Greetings, Rick. We have a fantastic presentation. I was curious about the hypoxic environment during osteoarthritis. All the effects on the cartilage, .L1, seem to be downstream of the modification of the environment of the oxygen. Did you look at the synovium and look to see how .L1 is modified in this tissue? And if the inhibitors you used for therapeutic modification were modifying this tissue? Could OA simply be solved by modifying vascular invasion into the synovium, thus restoring the homeostatic hypoxic environment?
Many questions in one well-elaborated comment, so it's fantastic writing. I would say we looked at the synovium, but the DMM model, those that have worked with it know that is a very low inflammation model. And we did not detect many differences or any consistent differences there. We haven't really looked or quantified the H3K79 methylation in the tissue there, so I cannot answer that one. Could the way simply be solved by modifying vascular invasion of the synovium? That may be a little bit, that may be a bit a shortcut, but it could definitely play a role. I also think that we could, and it's an idea that I contemplated already for a couple of years, that in the deeper layers where you have more vascularization due to the increased hypertrophy in the production of VEGF, that we could try to target this vessel formation out there in the deeper layers of the cartilage. But it's definitely an interesting idea.
Excellent. Thank you, Rick. A question from Nigel Haroon. Great talk as always, Rick. A couple of questions. First, where do you see notch signaling interacting with the proposed pathways? And secondly, recognizing there are very different forms of OA, DIP OA, hip dominant, post-traumatic, do you see a role for the HIF1 pathway as being the same at all or different phenotypes?
I think Nigel is taking revenge for all the questions that I've asked him over the couple of years, but about notch, I wouldn't know honestly to what to precisely answer. The whole system, of course, it's extracellular matrix, it's cells surrounded by an extracellular matrix. So transforming growth factor betas, bone morphogenetic proteins, notch signaling, Wnt signaling, they all interact at some level, and finding the balance, I think, is critical. Now, I have to say that as far as I remember out of the top of my head, I haven't seen notch signaling pop up in any of the systems that we manipulated. But these bioinformatics pathway analysis exercises, as many of you will know, they only teach you as much as what the input originally was. So I would not exclude the effect. And to the second part, yes, I started by saying that to me, osteoarthritis is an outcome of many different diseases. There are differences between hand OA, between the hip, between the knee. We are seeing emerging data about differences in the chondrocytes itself. Do I see it everywhere? I don't know. I think that what we mostly do in cell experiments and in mouse models, explants, is working on the type of cartilage that you find in hips and knees, larger joints. So I think there's a good rationale and there is good data showing the hypoxic environment. If you think about the DIP joints, we know there actually the DIP joints don't have the pure articular cartilage. It's more of a fibrocartilage. So it's it could be a totally different ballgame. And it's one of the many challenges that we obviously have.
So next question is by Guangzhou Zai. Nice talk, thanks. I wonder if you can comment on the crosstalk between Wnt signaling and TGF-beta signaling in osteoarthritis.
Yeah, again, not an easy one. I've worked a lot in the past on BMP, bone morphogenetic protein, and TGF-beta signaling, mostly in the context of axial spondyloarthritis, but also somewhat in the context of cartilage biology. The overall impression is that, for me at least, that TGF-beta signaling is doing a great job in keeping the joint healthy, but that in a pathological environment, that there are a number of switches that steer this pathway into a different direction. They do crosstalk, and especially in the healthy joint, I think they keep each other in check. In the pathological joint, because of the switch in TGF-beta signaling that has been suggested by the work of Peter van der Kraan, showing how the receptor is changing from the transition from the traditional receptor ALK5 to more ALK1 signaling. The pathway is differentially activated, and there they seem to work together in actually boosting a hypertrophy like chondrocyte phenotype, which I think is a deleterious event within the articular cartilage.
I had a question, Rick, about subchondral bone in these models. We know that obviously MRI studies in hip and knee OA showed a lot of MRI lesions, whatever that represents, and inflammatory lesions in the bone marrow. And I wonder, in the model of the ENP32 or the DOT1L knockout, are you seeing any changes in the subchondral bone marrow?
Yes. But we've seen some changes basically in what would say the composition in some of our models of the bone marrow. We have a few other genetic models where we see more lipid tissue there. We rarely see in our models an increase in classical inflammation in these bone marrow niches. And I still wonder, and we had the same question also in MRIs of the sacroiliac joints, at some point, what are you seeing? You're seeing the edema. That basically means fluid, but is this a full inflammation picture? I do think it is, because what you see on an MRI in OA, if you have a bone marrow lesion that is flagged as being inflammatory, and which is a predictor of poor outcome, it also looks a lot like what you see close to a fracture or close to a stress fracture. So I think there's an inflammatory reaction going on there. Yeah. But how that is connected to the subchondral bone remodeling with increased bone metabolism and the sclerosis that we can see, I still find that very difficult to understand. And our mouse models do not give us the answer, I think, at this moment.
Rick, in terms of the Wnt inhibitor, Semimed had, I think, SM04690, which probably is in the advanced clinical trials. Do we know the status of this, and if you have investigated this in your animal models?
No, we haven't used, but as I put in my disclosures, I am among the senior advisors on the senior advisory board of the company. So so the molecule has now an official chemical name, so it's no longer the SM and all the numbers, which are very hard to remember. So it's called lorcivivint. It has gone to phase 2 trials, and the phase 3 trial has been finished, and the results need to be finalized and then communicated. The phase 2 trial suggested a bell-shaped type of effect, which was more or less consistent with the concept of having not too little and not too much. They called it the Goldilocks zone, because that appears to be a very important fairy tale in San Diego, apparently. I had to look it up, to be honest, but anyway. So there was kind of, in the phase 2 trials, it was a bit of a challenge to find then the optimal dosing, but the overall analysis indicated some structural effect in that group, and that's been taken forward in the phase 3 trials. What the trials have, or the advisory position has taught me over time, is that it's really difficult to design trials in OA and knee-OA for many different reasons. What is your preferred outcome? What does the FDA and our side EMA want to see? Is it structure? Is it pain? How much time do you give it? So I think that the analysis and the communication about the phase 3 trial will be coming. I anticipate that it will be probably before or at the ACR, which will take place, if I remember well, in November, right?
Philadelphia.
That's wonderful, Rick.
A question here from Robin Poole, Rick. Terrific talk. Are there any signs in your studies of the impact of gender on these signaling mechanisms in view of the gender-related differences we see in OA?
Yeah, it's a great question. As Dr. Poole will know, mouse models work a lot better in male mice than in female mice, because in female mice you have to overactomize them first, otherwise the estrogens are way too protective. We haven't, well, so in terms of really functional experiments, we haven't challenged that we haven't done that in the female mice. We do have some ex vivo data from female animals that we use for some of the analysis, where we had the impression that at least the molecular signals went into the same way. My biggest question is, or my biggest, and at the same time my best reply to Dr. Poole is that, yeah, it's very difficult to take out the impact of the potential protective impact of female hormones in a model setting. And yeah, what we see in the clinic is, especially for smaller joints and for inflammation, that a postmenopausal status and the period thereafter is definitely something that plays quite a role. But I also agree that, or I also would say that the differences between gender are still understudied in many of the project proposals that that you see and studies that you see everywhere. Yeah.
I remember correctly, Rick, from a previous presentation, you have an interest in long distance running, and since there is a great deal of controversy about whether running is good for your articular cartilage in your knees or not, and whether it accelerates osteophyte formation, do you have any thoughts on what running actually may do to hypoxia and these mechanisms?
I'm not so sure it's about hypoxia. I think that, you know, gradual training, having good muscles, contributing to stability, they're all good things. So I think running, even long distance running, I'm convinced long distance running is in principle healthy. But you can outdo yourself. I can show you a brilliant MRI of my sacroiliac joint after a stress fracture, which you can use for differential diagnosis purposes. You know, injuries happen. I do think that you can generalize. If you have somebody who has a BMI of say 33 and they get the advice, oh, you have to exercise more to lose some weight, and they start running, they're going to put way too much weight on their hips and their knees anyway. So that's not and all of a sudden, that's not the best approach. If you gradually build up, I think it's good, and it gives a mechanical stimulus to your chondrocytes to make some matrix. So yeah, so I'm still on the positive side there.
Good.
All right, Rick, we have another question from Noah. Epigenetic modification of histones and DNA methylation state are associated. Have you looked at DNA methylation in your Wnt model?
The answer is no. We haven't invested into DNA methylation epigenetic screening. It's certainly not at a large scale. If I go back a little bit over a decade, we have one postdoc who was looking into a few target genes, but the evidence was inconclusive. But that was targeted with the pyrosequencing. I haven't followed the finer details of it, but when we were more involved with the GWAS studies, together with 328 consortium at the time, we were looking also into the epigenetic profile. And at that point, what we had available in terms of chips was giving us too little detail, I thought, to answer the question that we have. But since then, we've mostly looked from the epigenetic point of view at histones. I'm sorry.
All right. Rob, do you have any additional questions?
That's one question, Rick, because I was struck by your cardiac hypertrophy in one of the models. In the .L1 inhibitors, are you seeing any off-target toxicities that might pose a problem for moving this into clinical human studies?
No, we haven't. But yeah, we've worked mostly also within the intra-articular environment. So it's actually the opposite. So we need to restore. We need to maintain the function there. Yeah. There is a series. I think it's phase 3 or even in clinical use. This EPZ molecule that we use to inhibit it is close to the market, at least, for the treatment of a very specific subtype of leukemia. So there are a few subtypes of leukemia in which, but it gets a bit complicated, in which the mutation that is triggering the cancer development is linked to a translocation that makes that .1L is forming a complex with another molecule, and therefore goes to sites where it actually should not go. And that's the context where they developed the .1L inhibitors, which are very effective for this specific type of leukemia. But it's a different side of the spectrum.
Right. Thank you.
All right, Rick, we're right on time. Thank you so much for joining us today.
Thank you.
Thanks a lot, Rick. Stay well.
See you.
Bye-bye. Thank you.
Bye-bye.